Share my screen. So today we're going to do RNA-seq data processing and analysis, and we are going to do everything from command line. So first, what we're going to do, we're going to show you like genomic alignments. So when we say genomic alignments, it's included both genomic and transcriptomic alignments. Okay. But when we say genomic alignments, it's going to use splice-aware aligners. So the other one, when we say only, for example, RSAM, it is only going to use align the things, the reads to only exons or genic regions. So for that, that's why we call this like transcriptomic alignment and quantification. So today we are gonna go over all these exercises, but we want to give all this time to you today that you can actually spend time and do the exercises yourself. But first, I want Elisa gave another talk about this genomic alignments, and she beautifully explained all these things. And I want to start with that first. And after that, we are going to do, I'm going to explain what we're going to do here. And you're going to start all these exercises by yourself first. And then we're going to go over. Okay. But I want you to read all these things here, understand yourself, and then follow all these instructions here and give us feedback, like if you don't understand anything. So let's start with this genomic alignments.
So today we'll start by talking a little bit more about genomic alignments.
Sorry, wait a second. Sorry. I don't know what happened to my Zoom.
Just to...
Oh, sorry. I lost my Zoom here. Interestingly. Guys, I don't know what happened to this, but I lost my control. Guys, could you check if I'm still host here? Because I cannot have the controls.
Let me see.
Yeah, you are the host, Alper.
I'm host, but the screen, okay, came back. Okay, interesting. Okay, cool. Sorry, guys. I'm gonna start this again. Share computer sound. I was trying to arrange the sound because sound sometimes is not going good, and I'll do that. Okay, so I'll start.
Recapitulate from last week. What we were looking into was after you have your sequencing done, your result are reads, which you now know are contained in FASTQ files. And we need to figure out where exactly in the genome they came from. So that's the mapping step we'll go over today. And now you know also that the reference genome for the mapping is contained in FASTA files. So you have all the bases to get this process started. All right, so after the mapping, we can generate the coverage profiles and start to interpret this information. So we'll go through the analysis that we mentioned for RNA-seq and for genome alignments and go into some details. All right, so first of all, let's define mapping and alignment. Mapping is the process of finding the region or regions that read sequence can be aligned to. So there can be more than one position in the genome where a read will be mapped to. And alignment is the detailed placement of each base in that read. So the optimal base by base alignment of the read sequence to your reference sequence. So for each mapped location, you can examine the alignment, for example, and observe if there are any mismatches, if there are gaps, if there are insertions, or if it aligned perfectly to the genome, which is not usually the case. All right, but what are the challenges? The challenges are that the genome, like we said before, is large. And as you can see here, it is composed by a lot of repetitive sequence. So I took this view from Repeat Masker. It is a database of repeated elements in the genome. And here, the black portion of this pie chart is showing the genome fraction that is not composed of repeats. And you can see that it's smaller than 50% of the genome. So that means that all the rest are different kinds of repeat elements that are spread throughout the genome. And that makes alignment really hard. So just another analogy to go through this, it's like we in the Garber lab, we love assembling puzzles. And I had a really hard time the other day that I was trying to assemble one that it was composed of snow. So this is just to illustrate, it's kind of similar to when we try to map something to the genome. You have this puzzle piece, this puzzle image, which is basically composed of 4 colors, blue, white, and so on. And you're trying to find a unique pixel in this image that should be the size of a building and to be an accurate reference to the genome. And it's really hard because you have multiple positions where you have that exact same color that's gonna, that pixel is gonna be a good match to. All right. So let's go through what do aligners do to be able to go through the large genome and to find the best match and not take a week to do so for all your 15 million reads that you generated in your sequencing run. So the answer to that is that aligners create indices. So what is an index? It's just like the index for a book. So it allows you to quickly find sequences that are contained in it. So we'll go through this example for a hash-based index, and we'll see if it becomes more clear. Let's say we have a toy genome. In this case, instead of 3 billion bases, we're only going to have 16, and this is your entire genome. If we were to create an index for this genome, we first have to choose what is the hash size, so what is the k-mer size for this index, and we chose 3. That means that we're going to create an index that is composed of 3-letter words. So the first word in this genome will be CAT. And we annotate in the index what are the genomic positions where we see this particular word. In this case, we only see it here and we don't see it anywhere. Well, the first time we see it is in position 1, and we just walk over one base and continue writing down the different words that we have. So the second word we find is ATG. We annotate it as position 2, and we go on moving one base at a time until we get to the end of this genome. But as you can see, whenever you reach a word that is repeated, so for example, we see a second occurrence of CAT here, you're also going to annotate it in the same position you had for the first one. So CAT can be found in positions 1 and position 7 of the genome. So this is a complete hash index of our toy genome. And now we can see how easier it's going to be to map some read into it. All right, so let's try some hash-based mapping. So we're going to use the index to find the alignment location for this read. So we got a sequencing run result, and our read is 6 bases long. It's this one here. We want to know where it matches the genome. All right, so first of all, we split the read into the same sized words as we used to construct the hash index. So the first word in our read is TGG. And now we look for in our index, where can we find a match? And we find a match here, and we write down the positions that possibly this read could be in. So based on TGG alone, this read could be starting in position 3 or position 10. And then we find TCA. So TCA is in position 6. We write that down here. But now that eliminates some possibilities. For example, with the combination of TGG and TCA, it's much more likely that this read came from positions 3 and 6, because that would make it contiguous, as opposed to it having a gap and being aligned somewhere else. So we found the correct mapping of this genome really easily by breaking it down, instead of having to match this read to every possible location it could have come from in that genome. All right, so what is a good choice of hash size? So in this case, we had a small genome example. We used a small word. But if you think about the human genome and you think about a K size of 10, so words of 10 bases each, you can have up to 1 million different words composed of 4 nucleotides that are 10 bases long. And every one of these words is present in the human genome at least once. So even with a k-mer of size 10, you will certainly find many different positions in the genome where a similar word came from. All right, so what I want to show with this graph is that it takes a long k-mer to be unique in most genomes. So what I'm showing here, for example, you can see this start around 10 bases. This line here represents 50 bases. And if we follow it up, you can see that with 50 bases even, you're not 100% guaranteed to find a unique position for your read in the human genome. The human genome is shown in blue. And with 50 bases, you would get about 92.5% of your words to have a unique position, but not all of them. So think about this in terms of the sequencing reads that we usually have. 50 bases is a very regular size we have. Not all of them are going to match uniquely to the genome. That's already to be expected. And if you do smaller sequencing where you have reads of 30 base pairs or even smaller, it's going to be even harder to find that unique mapping position. And some genomes are just weird. For example, this parasite Trichomonas, you can see that it follows a completely different pattern than all the other genomes shown here, where it would take a word of 800 bases to make all of them unique mapping positions to their genome. So that's how repetitive it is compared to the human genome. And it's actually a very small genome too. So that's really curious. All right, so we talked about hash-based alignments, but that's not the only way to do an alignment, to create an index, I'm sorry. So there are other ways to create indices, and some of them are very commonly used in mapping algorithms. So for example, we have this strategy called space seeds, where there are 4-letter words that are masked, and then masked 4 letters, and then again a 4-letter word, and all of this is stored in an index, and you first try to match the words that are not masked, and then later on a second step you match the words that are masked. So this is a good strategy for alignment, but it takes tens of gigabytes to store the entire human genome in this sort of seed index format. So it's not very memory efficient, and it actually consumes a lot of resources to do so. So a strategy that has been used with success and initially came from algorithms that are used to compress files is called the Burrows-Wheeler transform. So for example, Bowtie is based on the Burrows-Wheeler transform and is one of the mapping algorithms that we'll use the most. If you're curious about how it works, I suggest just giving a quick Google to the Burrows-Wheeler transform. I'm not going to go into the details, but it's basically you take a string. So in this case, your string is the entire human genome, and you create all possible combinations of it by switching one base, by rotating it one base. And after you do that, so you concatenated the whole human genome into one string, you created all possible iterations of it, the Burroughs-Wheeler transform will just take you order them lexicographically, and the Burroughs-Wheeler transform will take just the last letter of each of these and form a single string that it can use to reverse match and find that position in the genome. So it works in a really fast way, and the index is only 2 gigabytes large. So it's about the same size as the genome unindexed, and it still serves its purpose for quick searches. All right, so then again, after we map, we still have some uncertainty, and we quantify this uncertainty about where this read alignment came from with the mapping quality. So it's the same as the probability that we used for base scores before to evaluate if a base was a correct call or a wrong call by the sequencing software. But now it's going to represent the probability that the sequence should be mapped there and only there in that position of the genome. So mapping quality also uses a Fred log scale, which we learned for the base qualities. So again, the formula is -10 multiplied by the log of base 10 of the probability that you got the mapping location wrong. So in this case, if you have the probability of getting the mapping location wrong as 0.1, then that means that log 10 of that would be -1, and the mapping quality would be 10. So with a mapping quality of 10, it still means that you could have gotten it wrong. You have a 10% chance of having gotten that alignment wrong. So this is what we use to interpret the alignment in the end of the software. And looking at the distribution of mapping qualities could be interesting for troubleshooting, for example. You have a mapping run where 80% of your reads align, you're probably satisfied with that. But if you have smaller and smaller fractions of your reads aligning, then you're probably going to want to delve into this and see what's happening to my reads. Is there a problem with mapping qualities? Does this mean that maybe my sequencing library is not good and I should re-examine the quality of them? All right, so let's just recap what we went through. So the read alignment relies on this dictionary we created. So once it's loaded into memory, and now you see the efficiency of an index that is 2 gigabytes large, or an index that is 20 to 40 gigabytes large, you can process thousands of reads per second. And different aligners are going to offer different advantages. For example, we have a variance of sensitivity and variability. So you can align to highly variable organisms. It might require you to have different parameters or even a different aligner to begin with. You have differences in speed. So some aligners like HiSat and Bowtie is actually super fast, are going to offer different advantages in terms of speed compared to others that are super slow but maybe more accurate. So it depends on what you're looking for. Memory efficiency based on the indices mostly. Specificity, which comes at the cost of the sensitivity of your alignment. And this is by no means a solved problem. There's always new alignment algorithms showing up. So you shouldn't assume that the one you're using now is the same one you're going to be using 5 years from now, although some of them have become quite staples in our arsenal for pipelines, and they are still some of the best to solve this problem. And in the end, I want you to keep in mind that you really don't know which region was actually sequenced. All you know in the end of this process is what is the best match of your read to the reference. So that is the best guess you can make, and it's good for most applications, but it's not a guarantee that you'll never know which place in the genome your read actually came from with absolute certainty. So one of the factors that that leads to this is that there could be sequencing errors. So your reads are not the exact match to the same fragment that they were created from, because the process of sequencing them introduces errors to begin with. And you also have variability from the genomes itself. So you're aligning to a reference genome, and you have to think that that genome is not going to match the genome, the exact genome of the cell you're growing in the lab. So there's been several different mutations that have happened in the lab. And other than that, there's variability from human to begin with. So the human reference genome is a composite of several different humans, but it is to be expected to have several different mismatches to the reference genome, and that will impact alignment too. And lastly, so you will have non-unique alignments. That means you have positions in the genome that are repetitive, and therefore you will have multiple possibilities where that read came from originally. All right, so this is just to show you the abundance of sequencing aligners that are out there. This is a table from 2015, and it's just listing all the algorithms, and you have here what kind of properties that algorithm has. And this is just a piece of the table. It actually goes on for longer and longer, and it doesn't even contain all of the algorithms that are currently available, and they're actually used for a lot of different purposes. So let's just summarize that to the ones that we currently apply to more to our data sets. You have aligners that are specific for DNA-based reads. So reads that came from the genome to begin with, like ChIP-seq reads, ATAC-seq reads. You're probably going to use Bowtie, which is the one that uses the Burroughs-Wheeler transform index, like I mentioned. And it's good for finding mapping for single-end reads as well as paired-end reads, and even for finding structural variations if you have them in your data and if you have interest in finding them. And in terms of aligners that are good for RNA-seq reads, and I'll show you in a minute why they're different, you're going to use STAR today to align reads to the genome. And it creates an index that is hash-based. So it's like the example that I went through line by line, that creates an index like a book that tells you where each word is found in the genome. And let's go through why it's different to have aligners that are made for RNA-seq compared to DNA reads. So what is the RNA-seq mapping problem? RNA-seq comes from a large majority of mature transcripts, right? Which means that for genomic mapping, we need to consider that this transcript, where they originated from, has been spliced. So it's not going to be found in the genome exactly like this. So many reads will likely span more than one exon, and therefore we cannot align it to the genome in a single stretch. So how do we do that? So we have FASTA files that contain the sequence of the genome, and we have the annotation files that contain the positions of known exons of known transcripts in the genome. So an aligner that is hash-based can try to, for example, STAR, can still map these reads because it first tries to map the biggest stretch that it can from a read. And when it can't align anymore, it does a second alignment separately for the remaining stretch of that read. And in the end, you're going to have a correct alignment for your reads that are entirely contained in exons, as well as the reads that span more than one exon in the genome. And another possibility is that you can have an aligner that is not splicing aware. So this is what we call a splicing aware alignment. And you can perform that alignment using a transcriptome reference. So this is something you're going to do today too when you use the RSEM program for RNA quantification. That program is actually in the backbone using the bow tie aligner, and it's mapping your reads to a reference transcriptome. So what is a reference transcriptome? So just like the genome can be contained in a FASTA file, so can the sequence of each of the known transcripts. So this is a already spliced, mature sequence of each transcript you know you have in your genome, and it can be used for mapping the reads directly onto it. So now there's no problem anymore with reads that will map to more than one exon because the exons have been spliced computationally. All right, and the advantages to this process of mapping to the transcriptome, for example, are speed. So think about if the transcripts are about 2% of the genome, that means that you're aligning to a much smaller universe of probabilities. And therefore, the alignment will go much faster. You don't have to look for in the entire genome for a probable match to your sequence. But what are the disadvantages? The disadvantages is that it relies on having a quality transcriptome annotation. So if you're working with an organism that is not widely used, you probably don't have that available to begin with. And you're more likely to be stuck using the genome and then making inferences of where your transcripts came from. But there's also another issue, even for genomes where you do have a good reference annotation, and that is you will not discover potential new isoforms. So if the goal of your experiment is to find novel transcripts and novel isoforms, that cannot be done if you only inform your aligner of the transcripts you know. So mapping to the genome will maybe give you a new isoform that doesn't contain an exon, for example, or that contains an exon that was not known in this in the particular annotations that you have now. So it all depends on your goal. And then lastly, let's just go through this strategy again that these aligners do for mapping first the exon first spliced alignment. So softwares like Top Hat will first try to map all of the reads that it can that are contained wholly inside exons. And it knows where the exons are because, like I said, we feed it not only the genome FASTA sequence, but we also feed it a GTF annotation file that tells it which positions of the genome contain an exon. So it'll first map all the easy reads, let's call them. And then later, it will do splice read mapping where it knows all the junctions that should be there, and it even is able to find new junctions that you didn't know were there. And it maps part of these reads only, and it can stitch them together in the end and find the splice read mapping. All right, so I'll leave you guys with some references. You probably noticed that I used a lot of the slides, including the toy genome from Aaron Quinlan. And there's a lot more from his lab that can be found here in this link. And if you want to go deeper into the bioinformatics aspect of some of these problems that I talked about today, that's a good place to look for some videos even. And if you want to read more on the problem of mapping billions of short reads, this is also a really good paper, and although it's from 2009, it has aged very well. And this is the reference for the repeat masker. And now you guys will go through the genome alignment exercise, and we'll come back for transcriptome quantification.
Okay, so before we start for the next exercise, I want to tell you a couple of things. Any questions, by the way?
And give you a couple of tricks that how we are doing.
Sorry. Okay, so any questions? I'm just gonna look. Okay, I don't see any questions here. Good. So before before you start the exercises, what I want to do now is, let me share the screen again. So, where's that? Okay. So this diagram here is a typical RNA quantification pipeline that we are using. So when we are using Dolphin Next next week, that you're going to see, you're going to upload your files into our system or into the cluster and locate where they are in Dolphin Next. But here, what you need to do is like you need to transfer your files to our cluster system. That just think about that, like you need to have like 10 times more space to process the data that you uploaded there. Just use like du -h for the directory you uploaded the files, and then you can measure the size of the files you have there. Usually they are zipped files, like think like this, when they are zipped, we need to extract those files to start processing the data. And they take a lot of space when you unzip them, when you extract the files like that. So you might realize that when you're doing your homework a couple of weeks ago. So, but what we are doing after you upload your files, we are running a FastQC program and to check the quality metrics first. And then if everything is fine, then we start processing data. So here you're gonna see today, you're not gonna do this ribosome alignment, for example, today. You're just gonna focus on STAR and RSAM here, and you're gonna align your reads to transcriptome plus genome using STAR. And then you're going to use RSAM later to do transcript quantification. So RSAM also using STAR or bowtie 2 in the background while aligning the reads to the transcriptome. So you can actually define it. So if you like using STAR, you can tell RSAM to use STAR while aligning, but you need to create necessary index files for RSAM to be able to process your data and do the quantification. And basically what we want to do after you run STAR, what we want to get out of from STAR alignment is a BAM file. And then you can use that BAM file and upload it to a genome browser, either IGV or like your UCSC genome browser, that we're going to actually show IGV today. And I will briefly show UCSC genome browser later. But you can visualize reads and you can go to the gene you are interested in and look how your reads are distributed along the gene. Are they on the exons only? Are they on there are some intronic reads there? Like, you can spend some time on it. So I was spending a lot of time myself and all the other scientists after I prepared these BAM files, the alignment files, that they were spending like days and nights to see if everything is how they expected. And also, like, are there some other anomalies there in the data, or are there some other interesting things? They were just like browsing all the, like many of the genes, like spending day and night just to see something interesting, because, you are doing these experiments and you want to look, everything is fine. You have like, if it is RNA-seq, equally distributed, like uniformly distributed reads on these, on your genes. And also, if it is total RNA, you're gonna see some like shorter reads. For example, you can quantify tRNAs or microRNAs or some other small RNAs like non-coding RNAs. And the RNAs actually that if they don't have poly A, if you do total RNA-seq, then you can get those kind of other RNA types. But if you do only poly A selected RNA-seq library, if, there are two different protocols that you can use, that if you just use this poly A selected, then you're not gonna have, for example, ribosomal RNAs in that case much. So it's gonna be like 2 to 3%. But you're gonna only have the mature like microRNAs with poly A tail. So there are two types of experiments for RNA-seq discovery. So then what we do after we quantify the transcriptome, you're gonna get a like expression values per gene per sample. Then we produce a table, just merging different samples into a single table, and then perform some differential expression analysis to find differences between different conditions, for example. And today, actually, when you go from top to the bottom of this tutorial, you're gonna learn like how we are doing these things from command line, from the terminal. So here you have like getting started. You have you're going to connect to the cluster. You're going to get your like interactive node first before you start, and run this script here, session 1 sh, and this will bring you all necessary files into your directories. That don't forget, like don't skip any step here, okay? Like go one by one when you're doing the exercise, load your modules, and then go to that specific directory. This directory will come in here after you run this session 1 sh script, okay? So don't forget doing these things. If you're stuck anywhere when you're doing these examples here, these exercises, so ask us on Slack, okay? And then we are gonna, Onur, Arthur, and I will respond your questions, okay? And this time I'm not gonna go over, you're gonna do it yourself, and you're gonna stuck somewhere, the things are not going to work, and you need to read these things too. Please read all these explanations here, understand exactly what we are doing, and when you stuck, ask us, please. And first, what you're gonna do is basically you're gonna create the index files that will use this annotation file, GTF file, to understand where to splice junctions in the genes. And then when you get this index files, basically you're going to use this index file while doing the other alignment exercises here. And then you're going to prepare RSAM index files here for start using the same UCSC GTF file that is already in your directories here. But don't skip anything here when you're doing, okay? Like, for example, if you skip CD, Bootcamp, or ASIC MM10, you cannot create that index file because this should be in that directory and you should run this program in that directory. And after you create these index files, then check if you have them and check your tree structure, do your checks. And then in the next exercise, you're going to do your alignment, okay? So please start doing this. I'll give you like maybe you can use 20 minutes and then I can extend later. If you cannot finish it, I'll ask you guys, you're gonna raise your hands if you're ready to move forward. But start doing this and read, please, from the beginning to the exercise 2, okay?
Seek mapping, because I know some of you were having trouble wrapping their heads around it. So we're talking about paired end reads and how you can use this probable distance between them. And I know some of you were asking questions about the aligner and the fact that it has to deal with the introns. So I just wanted to remind you all that here, this example is showing a very short intron. It's just a representation. But in reality, you all know that introns can be hundreds of kilobases long. So you can look in any genome browser and see the actual size of introns. And then you can understand how for a mapping algorithm, it is hard if you have a read that is split and 20 bases of it are in this exon, and the remaining 30 bases are 100 kilobases farther away. So the way that these algorithms deal with it, if they are splicing aware, so it's not the same as having a short gap. An aligner such as Bowtie that aligns to the genome is still able to map sequences that are mismatching from the genome by a few bases, or even that have short insertions or deletions. What it is not able to do is split a read 20 bases and 30 bases, hundreds of kilobases apart. So that it will not do. And the reason we use splicing aware aligners is that they, first of all, they know the most likely positions of exons in the genome because you gave it the reference annotation for that genome if you have one. And they go, they try to align the most that they can of one, of a section of a read. And when they can't anymore, they will look for the remaining section either in the whole genome in case that it's a novel splicing junction that's not annotated. But if it is an annotated splicing junction, they already have the positions where it's more likely for that remaining piece of read to be in, okay? So the hash indexing will serve for any base in the genome. It will have every base in the genome, what words, where you can find those words. But it's not the same as the splicing aware. Splicing aware means it's able to handle large gaps, large, like hundreds of kilobases large. And it's still capable of taking that second piece and finding a new position in the genome that could be hundreds of kilobases long, away, that aligns the remaining of your read. Sorry, I hope I didn't confuse you guys further. Let me know if you have more questions. A lot of the questions you guys asked on Slack, I'd be happy to answer during the class too. So just raise your hand or unmute yourself and feel free to ask. All right, so let's talk about quantification now. So there's a few things to consider about the quantification experiment. So now that you have the mapping position, let's go back and think about where the RNA came from to begin with. So you had, let's say, these 4 cells that you took, you made your RNA-seq libraries from. And these cells have different transcripts represented here by different colors. And you have several different molecules for each of those transcripts. They'll be present in different abundances and they'll have different lengths. So what you have to start thinking about in the RNA-seq experiment is that you will recover more reads from transcripts that are longer. So the red transcript will end up being present in more abundance in the output of your sequencing run. And you will also generate more fragments from transcripts that are expressed higher, at a higher level. So what do we want to find with the quantification? We want to find the actual expected relative abundance of a transcript. So what is the expected number of transcripts for a given fragment or a given gene? It is a result, so the number of reads you capture will be influenced by the actual expression level of that transcript and the fraction of your gene in the sample. And it will also be directly proportional to the length of that fragment. So if you have a longer fragment, you're going to get more reads, and also if you have transcripts that are more abundant. And this is the quantity that we're after. We want what is the expected number of fragments that we actually had in the sample to begin with. So to do the quantification, we have to make some assumptions. And these assumptions are that you have a high library complexity. What does that mean? That means that the number of molecules you have in the library is much greater than the number of sequence molecules you actually recovered. So what you see, as you could see from that previous slide, circled were the reads that you captured, but the actual fragments of molecules that were present in your sample are much more abundant. So you only took a snapshot, took a sample of a much larger pool of molecules. And the second assumption is that because the reads are short, then the read length is much smaller than the sequence molecule length, the actual length of the original transcript. So what you're capturing in the experiment is a portion of that original transcript, which you fragmented and then read only a piece of it. So of course, not all the applications satisfy this. This is what we're saying for regular RNA-seq libraries, but if you have microRNA sequencing, then you cannot expect that the read length is much smaller than the sequence molecule length because microRNAs are short. So it has to be dealt with differently. And if you have small input sequencing, for example, single cell sequencing, you can also not assume for a single cell that the number of sequence molecules smaller is smaller than the number of molecules that were present to begin with. So we deal with it in different ways. We have other ways to get around the amplification problem. And I'm not going to go into it now. It's just I wanted you guys to keep in mind that not all the applications will satisfy these two assumptions we're going to talk about. All right, so what are the challenges for the quantification? So like we saw in the genomic alignment and transcriptome alignment as well, there is uncertainty in the read mapping. So the genes have paralogs that share sequence similarities, so it's not necessary that even if a read came from gene A, it doesn't have a match also to the location of gene B in the genome if these genes are related or share, for example, a domain. So it's hard to define a single true location for each short read to have come from. And even if you do have libraries that satisfy those two assumptions we just said, that are more complex than the number of reads you sequenced, and that the reads are shorter than the actual original transcript, you can only measure relative abundance. The key quantity that we're after is how many fragments were sequenced for each transcript. So the data you have is aligned reads, and now you want to count how many fragments you have of each transcript. So what you want is which transcript generated, what amount of transcript generated the observed reads you have. So this should be easy, right? I can just count all the reads that I got for a single gene. Well, not exactly, because like we said, the reads don't uniquely map, and the genes have different isoforms with overlapping exons. Also, sequencing has a 1% error rate, which also already introduces a little bit more uncertainty on this. And the transcripts are not uniformly sequenced. So for example, you have certain biases that are known, for example, for GC content, where a given transcript will not generate the same amount of reads as another transcript with the same length, but a different nucleotide composition.
Can I ask a question
Yes.
before you go on to the next
Yeah,
slide?
go ahead.
Can you comment more about why you only are able to measure relative abundance? Like, why can't you convert these reads to something approximating an absolute number of transcripts? You know, if you
I think
make some assumptions about how efficient your library construction is and what percentage of your library you're actually sequencing, why can't you extrapolate out to something that approximates an absolute number?
right. So you can never, even though you know a lot of the biases that happen, so a lot of the losses that happen during library constructions, and you know, for example, the number of cells that you put into the experiment, but you cannot guarantee an exact approximation. So if you have, you're sequencing from a pool of cells, right? And even on single cell, you have losses during the library preparation, and you have these biases that will cause different transcripts to be captured differently. So you can never go back, just like you can't be sure of which sequence in the genome originated that read, you can never be sure of exactly the amount of RNA you had in your original sample. What you can be sure of, well, more or less, given these problems and taking them into consideration in your quantification, you can always use the measures of other transcripts relative to the one you captured. So that's kind of the information we have to deal with. So you can, it's a relative abundance. You know how much to expect of one transcript given what you saw of all the others, but you can never get the raw numbers. I'll go through the rest of the quantification. If it's not clear, I'll come back later to this point.
No, that was fine. Thank you.
Okay. All right, so we can try to simplify this problem a little bit. So let's start by saying that we know the gene and transcript annotations. So we know all of them. There's no novel isoforms to be discovered. We know everything that should be there. All right, so now let's simplify it even further and say there's only a single isoform that we can obtain per gene. And let's make an even further assumption and say we only have one read to one transcript correspondence, which means a read that we observe means one full transcript in the cell and not multiple reads per transcript, which is how fragments actually generate reads. All right, so we have this theta of G, which is the relative abundance of each gene. And we have lowercase n, which is the number of reads that align gene G. And lastly, we have capital N, which is the sum of all the lowercase n's for each gene. So this would be your sequencing depth, or more specifically, the total number of aligned reads that you got in your library. All right, so in this case, when you simplify it like that, when a success has the success is the probability that you got a read, has the probability of theta, which is the relative abundance of the gene. And so what is the probability of obtaining lowercase n successes in capital N tries, which is the total number of reads you got? So this will follow the binomial distribution. So it can be written as the probability of obtaining lowercase n successes in given that you have the relative abundance of gene theta, and then that is equal to the binomial coefficient, which in this case is capital N choose lowercase n, multiplied by the probability of n successes, which is theta to the power of n, and that is multiplied by the probability of having n minus capital N minus lowercase n failures, which would be 1 minus theta to the power of n minus n. So this means that our best estimate of the true relative abundance of this gene G is lowercase n over capital N, or the sum of all lowercase n's. So the best guess at the abundance that we can make is just to divide how many reads for that gene we got by the total reads we obtained. So we wish it was that simple. Remember all the assumptions we made on the previous slide and see how we cannot get a true relative abundance even for this gene based on all the problems we have to deal with in this case. All right, but let's go over the quantification steps again that we did. We have sequence reads and we align them to the reference sequence. That can be the species genome or the transcriptome, like we discussed. And we measure the transcript abundance by counting the reads mapped to each transcript. So this is not going to be accurate when you have multiple isoforms or when you have paralogs that share sequence. And then if you want the true transcript abundance, we can solve a maximum likelihood of the observed mapping given the transcript abundance. So to compare across samples these quantities, the counts have to be normalized. And why is that? So first of all, the libraries will have different sequencing depth. You are not going to be able to control, even though you try, so with pooling accurate amounts, you're not going to be able to control that you sequence the exact same amount of each sample. So first of all, you have to control for that. Secondly, the sample composition might be different. Even though, again, you tried to control your input, you might not have gotten exactly the same number of cells you did in experiment 1 compared to experiment 2. So the most standard normalization, which we're not going to go into in this session, we'll leave it for session 6 when we're discussing differential expression and normalization, is transforming the counts you got for each gene into transcripts per million units. And I'll go into it later, but basically you should just know that this measure will take into consideration the transcript length and the sequencing depth, because it's going to put the transcripts into a relative abundance form. All right, so what are we normalizing and why are we normalizing? If I did my replicates and I took care of everything to be right, shouldn't I have like a perfectly correlating scatter plot with minor deviations? Sure, it'll look pretty good, but if I put a line across what would actually be an exact replicate, you'll see what the problem is with unnormalized counts. So as you can see from here, without normalization, the quantification would imply that all the genes, almost all the genes, are higher expressed in replicate 2, which is here on the y-axis. So this is why we use normalization. All right, so let's go back to our quantification problem and now revisit our previous assumptions. We started with, again, we're going to keep it simple and say we know all the transcript annotations that should exist. But now, instead of assuming that we have one isoform per gene, let's say we want to use isoforms. We want to know if there's differential expression for different gene isoforms. So one way to deal with it is to define only one isoform per gene. In this example here, I'm showing you a single gene, and that gene has 2 transcript isoforms, one composed of 4 exons shown in dark blue, and one composed of 3 exons shown in light blue. So how do we define one isoform per gene? There are 2 simple methods that used to be very used for this, and they're called the exon union method and the exon intersection method. So based on all the exons that you see for both these transcripts, you can make a super transcript that contains all the exons known for that gene. This transcript doesn't actually exist, but it's a combination of the 2 that do. Or you can use the exon intersection method, which in this case will use only the exons that are shared between the 2 different isoforms that you do know. So there are problem with, so these are 2 simple ways to deal with the multi-isoform problem, but they both, and they will be convenient for mapping and quantification, but they will give rise to other problems. So the transcript abundance measured by either of these methods will differ from the truth. The union will end up resulting in underestimated expression for genes that have alternative splicing. And the intersection is going to reduce your power for DE analysis because it's actually going to be getting rid of a lot of reads that could give you information and more power. And lastly, we're going to give up our assumption of one-to-one read-to-transcript correspondence. So now we actually know that different reads could have come from the same transcript molecule. And lastly, we still rely on the fact that the read fragments are short and one transcript, because the reason we give up that assumption is because one transcript will generate many fragments. All right, so the problem with these methods is clear. So how can we do a better approach? The better approach that is used by RSIM and several other methods that try to do a transcript quantification is to do this probabilistic method of isoform deconvolution. So the transcript expression method tries to determine exactly how many which transcript did these reads originate from? What is the probability that each read came from transcript 1 or transcript 2? So it involves assigning these reads to a given transcript. And it must capture the assignment uncertainty. So for example, for the light blue and the dark blue reads, because they map to exons that are unique to each isoform, there's no problem there. So you already know where they came from. Using the quantification from these reads, you can also inform your choice for the reads that are mapping to shared exons. So these are the reads that you need to assign to a specific transcript given the probability that you saw other reads from those transcripts. All right, so your objective is to get the transcript relative abundance, not the gene level, but transcript level. And you don't know where that alignment actually came from. And what you do know is that you have a certain number of fragment alignments. You know the actual transcripts that should be there, and you also know the fragment length distribution. So let's go back to the fragment length distribution idea. So if you remember that we just went through paired end sequencing and that you have read 1 and read 2 for each sample. So here I'm showing you two pairs of reads. So you have read 1 and read 2. And here for pair 2, you have read 1 and read 2. And they map to different exons of this imaginary transcript. So first you have to perform a computational splicing and compute the insert distance. So you're not going to calculate the distance based on the genomic distance, but you're going to calculate it once this transcript is processed, and because the majority of your mRNA will be mature in your sample. All right, so you have for pair 1, you end up with distance 1, and for pair 2, you end up with distance 2. Once you do that for all of your paired reads, you can estimate this insert size empirical distribution. So you have a range of probable fragment insert size that you actually observed in your mapping. And you do this for every isoform reconstruction. All right, so let's go through that more carefully. You're going to use that for probabilistic read assignment. So for example, here we have a pair of reads. They're mapping to the first exon and the last exon, and you have 3 possible isoforms. And you need to figure out which isoform they actually came from. So for each isoform, you calculate the distance. For green and purple, the distance would be distance 1. And for green and brown, it would be distance 1, and for purple you would have distance 2, a shorter distance, as you can see here, because it doesn't have this middle exon. All right, so according to our distance size distribution, which of these is more likely to have given origin to this pair of reads? So based on this distribution you calculated, you see that it's much more likely that this pair of reads came from form 3. And this is the basic strategy that methods like meso cufflinks and RSM rely on. And because of that, it is critical that you have paired end data for this kind of analysis. All right, so we have this method for isoform deconvolution. The parameter that we want to estimate is the relative abundance of the transcript. And the variable that we don't know is the actual fragment alignment source. But what we do know is the number of alignments, the transcripts that actually exist in the sample, and the fragment length distribution. So the probability of a given alignment A to originate from transcript T, given the distance distribution D that we calculated, and the variable that we don't know, the parameter, which is the transcript relative abundance, is going to be equal to the fragment abundance multiplied by the transcript length divided by the sum for every other transcript that alignment maps to, that read maps to, of the fragment relative abundance multiplied by that transcript length, and that will be multiplied by the probability that given alignment with the length of L between the two reads is originating from transcript T given the distribution D. So uh, this is a problem that is solvable by an algorithm used by all of these problems called an expectation maximization. And what it will try to estimate is what is the transcript relative abundance, which will maximize the likelihood of obtaining the data that you did. So it will try to estimate this parameter we don't know, the relative abundance of the transcript, given the distribution of fragment lengths that are possible and the alignments that you got. All right, so to summarize, I just wanted to show you that the current quantification models are complex. They take a lot more into consideration. It's not just counting which reads align to the genes. And in its simplest form, we can assume that the reads are unequivocally mapped. We only have one origin from them. And then in that case, you could use the binomial for a single gene because the read counts will distribute multinomial for several genes with abundance estimated from the observed counts. But when this assumption breaks, then the multinomial is no longer appropriate. And the more general models that take care of additional information to accommodate the uncertainty of the data will use the base quality score. So it will inform how good an alignment is given a base quality score. It'll use the mappability of the sequence or how many alignments it got all over the genome. It'll use the information of protocol biases even, or if there is a certain trend to get more reads from a certain region of your genes. And it will use sequence biases. For example, are you more likely to get fragments from regions that are GC rich or GC poor? So handling each of these additional variants involves a more complex model, and the reads are assigned probabilistically. So not only to a different isoform of the same gene, but even for a different locus. So genes that share similarity in their sequences will also get a corrected count based on the probability that the read came from each of them. All right, so what does it look like in the end? You got through all of this and you have your results. So the genes are quantified, and each gene or isoform is going to have a fragment count value. So that can be the expected count, which comes from these probabilistic models such as RSEM, or the raw count, which is just a count of how many reads align there with no probabilistic model. And you also are going to have a normalized value, which we'll go into in another session, of TPMs, transcripts per million, or RPKMs, which I'll go into later. So all the samples are quantified in the same way and arranged into a table. For example, we have 22,000 genes that were observed in, for example, 6 samples shown here. And in this case, the table or matrix is going to give you, for each row I, all the expression of a single gene, say MIR301, in all of your samples. And the column J will give you the expression of all genes, all of these and much more, the 22,000, in one sample J, for example, control replicate 1. So the last thing I want to go over before you guys actually do your quantification and get your results is that RNA-seq libraries don't come all in the same shape and form. So we've been talking about libraries that cover the entire transcript, which are the most common, but there's other methods to generate libraries that try to capture the end of the transcript instead of the whole transcript. And the way they can do that is by either fragmenting and selecting a specific portion of the transcript or enzymatically purifying. And the uses of them are, for example, if you want to annotate transcriptional start sites. So the Phantom project tries to annotate the transcription start sites for all genes, and they use the 5' bias libraries a lot for that. Another reason is if you want to annotate 3' UTRs. So these are also cases where you can use them if you want to, you just care about quantification and expression at the gene level. So let's say for a given project, you don't really want to find new isoforms, you just want to do a quick count of how much each gene is expressed. This is a very good way to do that, because the depth required for these libraries is a lot smaller, since you're sampling only a small portion of the fragment. And another reason to use them is for low quality RNA samples. Those are degraded to begin with, and therefore you would never get a correct coverage of the entire transcript. So using the end sequencing libraries could be very useful for that. And another reason is single cell RNA sequencing. If you work with the data, you know that it's 3' biased, or even some methods are 5' biased nowadays. It's much harder to get high throughput, full coverage for single cells when you want to sample more, a large number of cells. But for 3' bias libraries, it's actually quite simple. So this is just to illustrate these three schemes. You start from poly A selected RNA for all three of them, for example, and you get fragmented RNA, of which you can then select either the 5' end based on biotinylation of the cap, or you can select the 3' end based on oligos against the poly A. And you can also sequence the full length, which will capture all of these regions of the transcript. So what does it look like? Since the next part of this session, we'll be talking about visualization. Here is how a coverage data for full-length RNA-seq library would look like, where you have in regions of exons, you have a higher coverage and they distribute across it, the reads distribute across it. But with 5' bias data and 3' bias data, here I'm showing you for a single gene, IL-23A, you would have only a pile of reads in the very 5' region or in the 3' UTR of that gene. And this is what it would look like across all genes in your sample. So because genes have different sizes, we just normalize this by gene length. So instead of using the actual length of genes and bases, we use bins. So you see that the accumulation of reads in the 3' bias libraries happens in the final end of the genes. And the accumulation in exocage data, for example, happens in the 5' end of genes. And I'll leave you guys with some references if you want to know more about methods we have for quantifying RNA-seq and their problems. And if you want to go into the problem of using expectation maximization to do probabilistic read assignment, this is a great paper. It's by the group that developed RSEM, but this is just the reasoning behind that method. And lastly, I'll leave you with this other link of more resources to think about how to construct your RNA-seq libraries based on the purpose that you want to use them for.
All right. I will continue with today's session. So over here, you came to the point where you executed your commands with the star. You build your star index and you do the alignment. Now we will continue with the RSEM. You already created the RSEM indexes, and now you will do this counting operation, transcriptome quantification operation, with this command, RSEM calculate expression command. So here is the details of each parameter of this command. And this is actually the command that you're going to execute for each pair of reads that you have. And this should create these results. For for example, for this one, it creates gene count values in this file, and in this file, it creates isoform counts. And also, you will have the spam files. And after you've done this part, you can check the structure of these results. They look like that. And please read all these explanations. And after that, you will execute this command, this rsm generate data matrix command. This will consolidate all the data that it's created from each pair of files, and that table gonna look like this. So for each gene, you will get a count value for each sample. So it will look like a matrix, and for just one gene count value, it will look like this. And it will be on Slack right now. So again, this might take about 20 minutes. And so let's meet around 11:10. And if you have questions, I will answer at that point, okay? We will be on the Slack right now.
Guys, we can add another 5 minutes if you need a break as well, but I guess it will be 20 minutes until 11:10, so it's up to you.
All right. Do you have any questions about this part? Basically, what we have done is we created a matrix filled with the count values. So the final file was located in this directory, Bootcamp, RNA-seq, quant, and the RSM gene summary count file going to be created. So if you look at that file, it's going to look like this. So each file headers, each file name is at the top, and they are tab separated like this. And for each gene, you can see the count values as in this table format. So the next step that we're going to do is we will visualize these alignment results. We will load BAM files into IGV. First, in order to do that, you need to download IGV. If you haven't done, please do that. And again, about this visualization, we have a short video of Elisa. I'm going to play this part, and I will continue with this visualization after that.
All right, so once we have our alignments, and even before we have our quantifications, we can use that data now for visualization. So there are a couple of options in terms of genome browsers, and I'm going to introduce you to one of them today, which is the IGV, the Integrative Genomics Viewer. So the reason we like IGV is that it is a desktop application, and it serves for you to visualize and interactively explore genomic data of any kind. It can be derived from microarrays, from epigenomic studies, from RNA-seq. You can actually see the alignments and the sequence behind each read. And you can do comparative genomics and look at the conservation of bases across different species. All right, the advantages of having a desktop app is that you run it on your computer and not through a web browser, like, for example, UCSC Genome Browser, which I'm going to give you a link for after. And you can input your own data easily. It's just a viewer. It's to make your visualization easier of this kind of data. So it supports any kind of data that can be mapped to the genome, and the input data that you can submit to it can either be stored in local files in your machine, or it can be stored in a server. And you can give it even an URL to access that file. So you don't necessarily need to download files to your computer just to input them to IGV. One of the things you can use it for is to visualize the actual read alignments that came out of your mapping step. So this is an example of an RNA-seq view of alignment. So each of these boxes represents a read, and actually multiple reads are seen here. And I'll point out two things. One is that for a specific read, you can see the gap between exons. So it will show this thin blue line when a single read is split. So it mapped partially to one location and partially to another. And you can see that here. And another thing you can see is that this RNA-seq library is the strand-specific library. So you actually know which strand in the genome your reads originated from, and that is according to the orientation of the gene that originated the transcript to begin with. And you don't use it only for RNA-seq alignments, you can use it for multiple kinds of alignments, and you can input multiple different libraries to a single browser view. So for example, here, I actually have 3 time points of RNA-seq that I'm looking at, and now I'm looking at only coverage tracks. So only the depth of coverage and how many reads were mapped to that position, not each alignment by itself. Here you have ChIP-seq alignments for histone K4ME3. You can have ChIP-seq data for specific proteins like Pol2, and you can have ChIP-seq for other transcription factors, which will all give rise to a different kind of broad or narrow peaks. And it helps a lot to evaluate the quality of the data when you have nice clean peaks for a given transcription factor and not broad peaks. But in terms of Pol2, for example, you do expect it to be all over a given transcript, so the data is indicating that you have good quality. And you can use it to troubleshoot. For example, do you expect to have, you can create this junction track, and you can see if most of these reads coming from other areas, not the transcript, are in a different strand and can be coming from enhancer transcription. And you can check for the splicing and see if you expect to have all of those splicing junctions given the isoform you know it's there. And here is a slide that I took from a course of this bioinformatics.ca portal, which is very good, and I encourage you guys to go through their material if you want to know more. And it's just to give you an idea of how the IGV screen will look like. You're going to learn to navigate through it. Here's where you're going to choose the genome that you want to visualize. And in this position here, you can input the location in that genome you want to see. You can use either a gene name or a specific location. And it should be, if you're using a location, you can give it the chromosome address and it'll take you to the entire chromosome. Or you can give it an actual starting or ending position. All right, and in the top here, you're gonna see this ideogram, which is just a representation of the chromosome with the bands. You have a red square, which will go wider or narrower depending on the zoom that you have. And it'll show you where in the chromosome you're located. So it's just a reference for you to find yourself. Here's where you control the zoom, or you can actually say the start and end of the position you want to see. When you load a BAM file, this is the view you're going to get. You're going to see the alignment read by read, and you are also going to see the coverage track on top. And if you go through the exercises, you will see what we ask you to do with these. So you can see, for example, there are single reads that are not spliced. And like I mentioned, there are multiple reads that are spliced across exons. And you will see them with the blue string connecting them. So these are stitched reads. You have here on the top of the coverage and any other kind of histogram kind of track you load, you're going to see a scale. So from 0 to how many reads you observed in that region. And this is where it'll show up the name of the read track you're looking at. So if you load different samples, it's going to show up here with different names. And in the bottom here, you have a gene track that will tell you the annotations that the browser knows for this particular genome. And you can change this track too if you want to modify it or add a different annotation to the one that's default there. So that's pretty much it. You guys can go through the visualization exercises we made for you. There's an IGV YouTube channel with lots of tutorials if you want to go through. And there's more course material in this bioinformatics CA with slides about each different kind of activity and things you can do in IGV. And if you want another browser choice with more public data available and already preloaded that you can just select and visualize quickly, you can use the UCSC Genome Browser. The reason I use it is a lot of the times I don't want to see my data, but I want to look at a particular region of the genome and see what the context is, if there are repetitive elements or anything like that. So Genome Browser is really good for that when you're not loading your own data. And that's it.
All right. If you want to reach the slides of Elisa, you can click these links. And at the end of the video, there are some great tools that you can find. You can check them as well. So if you already downloaded IGB, now you can go to FileZilla software and copy and paste the files that you created, the sorted BAM files and the index files, bam.py files, to your computer. So basically you will go to FileZilla and enter the ghbcc06umassrc.org over here as a host, and your username and password you will enter. Then you can click the quick connect. It will connect, and you will see the content of your cluster directories. So over here for the RSAM, for the star alignment, we have this sorted folder. If you just drag and drop this sorted folder to your computer somewhere you can reach, you can easily load to IGV. So when you go to the IGV, the first thing that you will do is choosing this genome that you're going to use, which is mouse MAM10 for our case, and then you will click load from file and go to the place where you copied your files and choose them like this. I'm choosing only the BAM files, but IGV will load the index files as well, but I'm not choosing index files while loading. So now it loaded, but it's in a kind of a zoom out version. So I can enter either a chromosome name or some gene name that it might be interesting. For example, this HGF 21. And it will zoom to that region. So as you can see, the reference RefSeq genes structure from the GTF file is located here. And then all my read information is located here. And I can compare the, by the way, these are the RSAM results. Sorry about that. I said star, but these are the coming from the RSAM results. So you can compare the expression levels. So controls, these three are the controls, and these three are the experiments. You can compare those results by checking these heights. And so what else you can do? You can load both star and the RSAM results and compare how they look. And also you can do other exercises over here. After that part, we will have this differential expression analysis. For that one, we will use this EB-seq in RSAM. It's a easy way to do a differential expression analysis. If you execute these commands, it will create these differentially expressed gene file for us. And for this exercise, it just showed that 11 genes and transcripts reported as differentially expressed. So you can check their results and the meaning of these values. You can check over here. And I will give you about, again, 15 minutes for this. And then we will finalize this session. Yeah, if you have questions, please ask. And we will be on Slack right now. All right, do you have any questions about this part, final part? So if you have any trouble with connecting FileZilla, you might try entering the port value 22 as well. Sometimes it works with that. And please try to do this homework. And when you're done, you can send your screenshots to our Bootcamp Slack channel. And if you don't have any questions, we can end this session. Thanks for joining.
So maybe I can just close too. So please go over all these things here by yourself. If you stuck anywhere, but you have to start from scratch. If you stuck anywhere, if it is not working, it means that you skipped something there. So don't skip anything, and try to check which directory you are on, like with using pwd command. And if you have questions, please ask on Slack to us while you're doing these things in the weekend. So hopefully we are going to upload the video too about today. And if you have questions, maybe on Tuesday morning we can go over your questions too. Thank you for joining today.
Alper, can we also can I interrupt you for a second?
Please.
Can we also repeat that announcement of ours to ask people to check their project spaces?
Do it. Can you tell?
Yeah, sure. I can actually tell people briefly, then we can go over again. Hold on a sec. I'm just going to take a few minutes of your time to just discuss this really quickly. Let me share. Where am I? Yeah. So guys, is everybody seeing what I'm seeing now? The terminal? Can you guys confirm? No? Okay.
Yeah, yes.
Okay, so basically now we've been working in the home directory. So basically it's the path that starts from your home and it's followed by the directory that is specific to your username, right? So there are other places that you have more space to do more quantification and alignment, especially when your sizes are much higher than the simple examples that you have over here. But for the last 2 sessions, when we cover single cell analysis, we will deal with much larger sizes of FASTQ files and how to align them. Alper also will show you how to use Dolphin next in either Tuesday or Thursday next week. And we're going to cover that as well while doing single cell analysis. And we need a little bit more size. So basically, some of you may have specific large project directories specific to your lab, right? So in order to do that, you should go to project instead of home. And starting from UMW, your PI and your lab should have a specific directory. So I am officially from Manuel Garber Lab here in Bioinformatics and Integrative Biology Department. So my space will be UMW_Garber Lab. So your lab should have a space like that too. If you go there, all the members have their own spaces. Here I opened a directory of my own. And you can just open a directory with your name and just toss stuff here. So basically, this space has minimum 2 terabytes, and we hope to work over there for our single cell analysis. So that's what I was planning to announce you today to you guys. And if you have it, it's fine. But if you don't, just tell us, let us know, and we can walk you through before the 7th and the 8th sessions. Did I miss something, Alper? Onur?
No.
Okay. I'll make the same announcement in Slack as well, and we can talk over there too.
Okay. Thanks, Arthur.
No problem. I'll
Okay,
stop
then.
sharing now.
Okay, perfect. Then let's meet on Tuesday to continue the classes. Then this was the hard way, by the way. You're gonna do the easy way on next Tuesday and Thursday. Okay. Talk to you later. Bye.